Hello everyone, welcome back to another video of STEMAC, and today we're going to talk about enzyme. So imagine if the chemical reactions that are taking place inside our body took centuries to complete, it would be a disaster, right? But luckily we have enzymes. So what are enzymes? Enzymes are biological catalysts. Biological because they are proteins with a unique shape, and catalyst because they speed up the chemical reactions inside living organism they speed up the chemical reactions, and so they are called catalysts. So enzymes are highly specific. They are specific for both products and the substrates. Also, they are not consumed at the end of the reaction, and they can be reused multiple times. So let's see what enzymes are made of, but before that we're going to talk about the classification of enzymes based on shape. So an enzyme can be of two types based on their shape, which is simple enzyme. So simple enzyme are mainly, they are protein as a whole, and a complex or hollow enzyme. So a complex enzyme has two parts. Apoenzyme. This apoenzyme is the protein part of complex enzyme. So they are the protein part, and this apoenzyme is inactive on its own, and they require a cofactor to be fully functional. So this is the protein part of a complex enzyme, and the cofactor part of complex enzyme is the non-protein part. And the cofactor attaches with a apoenzyme of a complex enzyme and activates them. So the cofactor can be of two types, which are prosthetic group and coenzyme. The prosthetic groups are the little inorganic molecules or metal ions, such as magnesium, right? And coenzymes are large organic molecules like FAD, NAD, etc. So these are the cofactors, and this is what this is the types of enzymes that we get based on shape. So in this figure we can see that here it is this figure of a complex enzyme, and here we can see that this is an apoenzyme, which means that this is the protein part of a complex enzyme, and with that we have a cofactor, which is the non-protein part of a complex enzyme, and together they form hollow enzyme or complex enzyme. So we can see that the cofactor has attached itself with the apoenzyme, and it has activated the enzyme, and this is our substrate, which will then bind to the enzyme. So how does an enzyme work actually? So before that we have to know what an active site is. So active site is a cleft on the surface of an enzyme. So this cleft is an active site of an enzyme. And a substrate molecule will bind to this active site of an enzyme. So together they will form an enzyme substrate complex, which is a temporary complex. So earlier we learned that enzymes are made up of proteins, and we know that proteins are made up of amino acids. So this structure here is a structure of an amino acid, and we know that the amino acids have an R side chains, right? So these R molecules at the active site will interact with the substrate, and this interaction will break apart the substrate into products. So as a result, from substrate we get products, and we get enzyme product complex, which is also temporary. So after that the products will leave the enzyme And as we can see that the enzyme is still unchanged from the beginning. So it has not changed and it can be reused for a new substrate next time. So this is basically how an enzyme works. And to put it in simpler terms, we say that the enzyme and substrate together, they will form an enzyme substrate complex. From enzyme substrate complex, we will get an enzyme protein complex, and from that the enzyme product complex, and from that we will get the enzyme in an unchanged form and the products. Now we are going to talk about how the enzyme perform this mechanism in their modes of actions. So there are two modes of action through which an enzyme completes its or perform its mechanism. So first is lock and key mode or lock and key model. Now lock and key model suggests that an enzyme, the active site of an enzyme, has a very particular shape, and only the substrate with a complementary shape to that active site can bind to it. So here the substrate is acting like a key to fit a lock, right? And here the enzyme will act only on one type of substrate, because only the substrate with the correct shape will fit in it. So it's very rigid, right? So in this figure we can see that we have an enzyme and it has a very particular shape. The active site, this is our active site, and it has a very particular shape, and this is our substrate, and the shape of this substrate is complementary to the shape of the active site of the enzyme, and this substrate will bind to the active site of the enzyme to form a enzyme substrate complex. So this is the lock and key model or lock and key mode of an enzyme. Next we have induced fit model. Now in this case it's kind of similar to lock and key, but here the enzyme or sometimes even the substrate can slightly change their shape to maximize the fit. In this case, the enzyme will still show specificity, but it will be more flexible than lock and key. So in this figure we can see that this enzyme has a particular shape. The active site of the enzyme has a particular shape, and this substrate, the shape of this substrate is different from the shape of the active site, right? So what's going to happen is that the enzyme will slightly change its shape to fit the substrate, and then the substrate will bind and form an enzyme substrate complex. So this is the induced fit model of the mode of action of an enzyme. Now with all this mechanism, enzymes do a very interesting thing, which is they speed up the rate of the chemical reaction, which we talked about earlier. But how do they do that? They do that by lowering the activation energy of a reaction. So what is activation energy? Well, activation energy is the minimum energy required for a reaction to start. So it's like an energy barrier, and a reaction has to cross this energy barrier, or otherwise the reaction will not occur. So in this figure, in this first one, we can see that here is an activation energy or energy barrier. Now this substrate, it is not bound to an enzyme. So this substrate will have to cross this barrier to make products. However, in this second figure, we can see that the activation energy barrier is much lower than the first one, right? Because in this, the substrate is bound to an enzyme. And because of this enzyme substrate complex, it will be much easier to turn the substrate into products, and so less energy is required, and hence the activation energy barrier is much lower, and this will take less time for the reaction to be completed. Move on to factors that affect the action of an enzyme. So first we have temperature. Now, if the temperature is very low, the action of an enzyme will also be low. That is because at low temperature, substrate do not collide at the active site of an enzyme, and so the enzymatic action will be much lower at a low temperature. Similarly, at high temperature, the enzymatic action will also be lower, because at a high temperature, the enzyme will denature, and that is because the bonds that hold the shape of an enzyme will start to break at a high temperature. So, the enzyme will lose its shape. Now, there is a temperature at which an enzyme will work their best, and that is called an optimum temperature. So, here we can see that we have an optimum temperature. In this figure, we can see that at low temperature, the enzymatic action is near 0, right? And as the temperature increases, collision will happen much frequently. So, a substrate will collide with the active site of an enzyme. And so, as the temperature increases, it will reach its peak at the optimum temperature. And at this peak, the enzyme will work its best. But when the temperature will cross over this optimum temperature, the enzymatic action will start to go down again, because at high temperature, the enzyme will denature. Now, different types of enzymes may have different types of optimum temperature. For example, human enzymes have an optimum temperature of 40 degrees Celsius, whereas enzymes found in bacteria living in hot springs will have an optimum temperature much higher. Similar to a temperature, enzymes can also have optimum pH. So, most of the enzymes have an optimum pH of 7. However, it may vary, like for example, the pepsin enzyme found in our stomach may have an optimum pH of around 1.5, because the stomach, environment in the stomach is very acidic, right? So, in this figure, we can see that at a low pH, the enzymatic action is very low. Also, similarly, in high pH, the enzymatic action is also low, but at this peak, it is the optimum pH. So, here the enzymatic action or enzyme is working its best, right? Now, why does this happen? Now, we know that pH is a measurement of the hydrogen ion concentration in a solution, and hydrogen ions are positively charged, right? So, hydrogen ions are attracted by negatively charged ions, and they are repelled by positively charged ions. So, sometimes earlier we learned that the active site has R molecules of the amino acids, because enzymes are made up of amino acid. So, they have R molecules at the active site. So, hydrogens will interact with any charged R molecules, and they will change the shape of the enzyme because of that. So, because of that, substrate will not be able to bind to the enzyme, and as a result, the enzymatic action will be much lower. So, in low pH, we know the concentration of hydrogen is much higher, and the hydrogen ion will react with the R molecules at the active site, and as a result, they will change the shape of the enzyme. So, therefore, the enzymatic action is low. Now, next we have enzymatic or enzyme concentration. So, the more the enzyme, the faster the reaction. So, if we add more enzyme and we have substrate that are still not bound to the enzyme, then reaction will be a very faster rate at the beginning. However, if we keep adding enzymes but the substrate is constant, in that case the reaction rate will not increase, because the enzymes are already occupied by the existing substrates. So, adding more enzymes will not give any effect. Similarly, we have substrate concentration, and the more the substrate, the faster the reaction. So if there are unoccupied enzymes and we keep adding substrates, the reaction rate will be very fast at the beginning. But again, if the enzyme concentration is constant and we keep adding the substrate, it will not put any effect in the reaction rate, because when all the enzymes are occupied by substrate, adding more substrate will not increase the rate. So now there are some molecules, other molecules that can also slow down or stop enzymatic actions, and they are called inhibitors, enzyme inhibitors. They are of two types, competitive and non-competitive. So in the case of competitive inhibitors, they bind to the active site of an enzyme. This is our active site. And they bind to it, right? So this is our competitive inhibitor, and we can see that it is trying to bind to the enzyme of the active site of the enzyme, and this is our substrate. So because of that, the substrate cannot bind to the enzyme anymore, and this will slow down or stop the action of an enzyme. So there is a constant competition between the competitive inhibitor and the substrate. That is why it is called competitive inhibition of enzyme. Now this can be reversed by adding more substrate than the competitive inhibitors. In that case, the substrate will win over the competitive inhibitors, and they can bind to the active site. So it's a reversible process. Next, we have non-competitive inhibitors. It is another type of inhibitor, but in this case, the inhibitor will bind to another site of the enzyme instead of the active site. So here you can see, if this is the active site, it will not bind to the active site, but rather to a different location of the enzyme. Now, because of this, they can affect the bonds that hold the shape of an enzyme, and with that, the enzyme will lose its shape and the active site will also change. So when the active site is changed, the substrate can no longer bind to it, right? And with this method, they can cause the inhibition of an enzymatic action. And because of the reason that they are not fighting for the active site directly, it is called a non-competitive inhibition of enzymatic action. Now in this case, adding more substrate will not overcome this situation, because here the active site is already changed. So when you are adding more substrate, it will not work. So this is all about enzyme, and we're going to end today's video here. Thanks for watching.